All right, hello everybody. Welcome to this, your first biology test. We're curious to see how well you do. You have 2 days to do this, so take your time on it. Here we go. So, document one is going to be used to answer questions one, two, and three. So, document one, let's study its features. You see cell, chromosome, DNA strand, gene, and DNA building blocks called nucleotides. In this diagram, you see sugar phosphate backbone and then the word gene. Remember, document one are these two diagrams, and they can be used to answer questions one, two, and three. Question one, what is the hereditary molecule contained in all cells? Number two, DNA contains small sections called that code for proteins. Question three, what makes one gene different from another? All right, document two. Here's a short reading. This reading is used to respond to all of these prompts. Here's the reading. The double-stranded DNA that makes up the the double-stranded DNA that makes up the human genome can be subdivided into information sequence called genes. Each gene encodes a unique protein that performs a specialized function in the cell. The human genome contains about 21,000 genes. Cells use the two-step process to read each gene and produce a string of amino acids that makes up a protein. The basic rules for translating a gene into a protein are laid out in the Universal Genetic Code. The first step is called transcription. In the nucleus, the cell's machinery copies the gene sequence into a single-stranded messenger RNA, mRNA, a molecule that is similar to DNA. Like DNA, mRNA has four nucleotide bases, but in mRNA, the base uracil replaces thymine. The second step is called translation. During translation, the protein-making machinery, which is located outside of the nucleus called the ribosome, reads the mRNA sequence and translates it into the amino acid sequence of the protein. The ribosome starts at the sequence AUG, then reads three nucleotides at a time. Each three nucleotide codon specifies a particular amino acid. The stop codons tell the ribosome that the protein is complete. All right, question one. Why is DNA not directly used in translation? In other words, why is DNA being transcribed into mRNA? Two, what are two ways a strand of DNA is different from a strand of mRNA? Three, why is it important to have a start codon and a stop codon in the strand of mRNA during translation? Four, sometimes a mistake is made during DNA replication and the wrong nucleotide is added. How could this impact the process of how a protein is made? All right, document three. Sorry, document three. This document is going to be used to answer questions one, two, three. There we go. Please notice document three has a title, Common Human Proteins and Their Functions. First, insulin, enzyme for sugar metabolism. Second, cytochrome C, enzyme for cellular respiration. Growth hormone, used as anti-aging treatment. Hemoglobin, oxygen transport in the blood. Hexokinase, enzyme for glycolysis. Gamma globulin, part of the human immune system in the blood. Myosin, involved in muscle action. And then in this diagram, you see this word undifferentiated cells. It says expressed gene, The number of arrows indicates the expression level. The arrows are directing down. Cell division specification. Cell differentiation. Nerve cells. Epithelial cells. So your job is to use everything that you know about DNA, DNA, mRNA, amino acids, and proteins to study this diagram and to use this diagram to answer questions 1, 2, and 3. Finally, document synthesis. That means using documents 1, 2, and 3 to perform the following task. Using evidence from the previous pages, construct an explanation for the following claim. You must use one piece of evidence from each document to demonstrate understanding. Claim. The structure of DNA determines the structure of proteins, which carry out the essential functions of life. What evidence do you have that supports this claim? How do you explain it? And you have to do that 3 times. Real-world applications. Use your codon chart to fill in the missing amino acids. And then finally, galactosemia is a disorder in babies that makes it so they cannot convert galactose into glucose, a major source of energy for the body's cells. Describe the mistakes you found in strand 2 and how they are evidence that the baby has galactosemia. So in strand 1, normal DNA for the enzyme that breaks down galactose. In strand 2, a sample from a baby's DNA for the enzyme that breaks down galactose. You'll probably notice an error as you fill this in. That is, something here is going to be different than here. Make sure you're using your codon wheel. So, and then finally, why is galactosemia a disease that can be treated but not cured? All right, students, do your best with all of these challenges. They are a part of today's test. Remember, you have 2 days to do it. Please reach out to Mr. Williams, Ms. Carr, Ms. Chick, or myself with any questions or support. Remember, our ultimate goal is to help you guys succeed. I hope you're all well. Goodbye.